Hello, this is Uzair Kashif, first year MSc Biochemistry. My topic is p53, also known as the tumor protein 53, cell tumor antigen or transformation related protein 53. So firstly, what is p53? p53 is a gene which encodes a protein that regulates cell cycle. Protein encoded by p53 regulates cell cycle and functions as tumor suppressor, hence it is also known as TP53 or tumor protein. p53 is referred as the guardian of the genome, as its role in conserving stability by preventing genome mutation. p53 denotes molecular mass of p53 as it runs as 53 kDa band on SDS page. Where it is located? So, it is located in the chromosomal number 17. Let's see what is the structure and function of p53 protein. The p53 protein is a phosphoprotein made of 393 amino acid. It consists of 4 units or domains. First one, a domain that activates transcription factor. Second one, a domain that recognizes specific DNA sequences, that is core domain. Thirdly, a domain that is responsible for the tetramerization of the protein. And fourth one, a domain that recognizes damaged DNA as a miselegant base pair of single-stranded DNA. Let's see how p53 regulates the cell cycle. p53 plays a major role in regulating or controlling cell cycle or apoptosis. In normal cases, level of p53 in the cell cycle will be very low and it is quite unstable, degrades quickly. When there is a DNA damage in the cell cycle, p53 gene expresses and produces is more amount of p53 tumor suppressor protein. Let's see the major functions of increased p53 expression and the gene regulations. First one, cell cycle arrest. Second one, DNA repair. Third one, apoptosis or cell death. Let's see how the p53 regulates the cell cycle. So the p53 regulates the cycling dependent kinases to the CDS and thereby inhibiting the S phase and M phase. And this is controlled by through P21, GAD, D45 and 14, 13, 3 sigma. So these are the regulating elements through which the p53 act and the cell cycle arrest occurs through this process. Let's see the role of p53 in apoptosis. So p53 can activate other pathways and through caspase 9. It causes the apoptosis or the cell death. So it is done through BAX and APAF1 and then through binding of the cytochrome C to the cap space and which causes the apoptosis. Let's see how p53 is regulated. p53 can bind to the MDM2 gene and causes the expression of MDM2 protein. The MDM2 protein can in turn bind to p53 and through ubiquitin mediated degradation pathway, MDM2 protein and degraded p53 is formed. So the ubiquitin will decrease the p53 MDM2 protein complex. But in case of DNA damage, p53 get phosphorylated through ATM kinase activation. Once the p53 is phosphorylated, MDM2 cannot form complex with p53. So no N53, so no p53 MDM2 protein complex will be formed. So this will further increase the p53 level. Because ubiquitin cannot decrease the p53, so when there is a DNA damage, cell cycle need to be arrested. So higher level of p53 is required So for that, phosphorylation of the p53 will occur through ATM kinase. Once it is phosphorylated, it cannot bind to MDM2 protein, and thereby it will not degraded by ubiquitin pathway. So the level of p53 will stay increased and stay in higher level. Once the DNA is repaired, ATM kinase is inactivated, and this will dephosphorylate the p53. Once the p53 is dephosphorylated, MDM2 complex will form through p53, and as a result ubiquitin act as a p53 MDM complex, and the level of p53 decreases. If p53 gene is damaged, tumor suppression will be highly reduced because this pathway will not be active. More than 50% of human tumors contain a mutation or deletion of the p53 gene. The degradation of p53 is, as mentioned, associated with MDM2 binding. In a negative feedback loop, MDM2 itself induced by p53. However, mutation p53 often don't induce MDM2 and are those able to accumulate at very high concentrations. Mutant p53 protein itself can inhibit normal p53.